Alright, let's start with talking about the genetic material, or DNA, inside of our cells. So, back when we first discovered that DNA was on chromosomes, we knew that chromosomes were made of two things: DNA and protein. But we weren't really sure in the early 1900s which part of that was the genetic information. Was it the DNA or the protein? The first attempt to show that DNA was actually the genetic material was in 1928 with Dr. Frederick Griffith. And he injected bacterial strains into rats and showed that dead bacteria could transfer DNA to living bacteria and go from non pathogenic to pathogenic. And so this was the very first evidence that DNA and not protein was actually our genetic code. And at the time, nobody really believed him. It wasn't until 1952 when Alfred Hershey and Martha Chase did another experiment showing that DNA was the genetic material that people started to kind of agree and go along with this idea. So you can see a picture at the bottom from the Hershey-Chase experiment. What they did in this experiment is these are viruses, these little alien-looking dudes, and viruses are made up of two parts. They have genetic code in their DNA, and they have a protein coat on the outside. And what we knew about viruses at the time was that they injected their genetics into bacteria. So what Hershey and Chase did is they labeled the DNA inside of the virus with radioactive phosphorus, and so it fluoresces green here. They let the viruses infect some bacteria, and you can see this one's inserting its DNA into the bacterium. And then they took these bacterial cells and the virus cells, sorry, not cells, virus coatings, and they blended them up and did a centrifuge. Well, a centrifuge is going to distribute things by density. So the heavier things form the pellet at the bottom, and the lighter things form what's called a superknit at the top. We knew that the bacterial cells would form the pellet at the bottom because they're much heavier than the virus coatings. So when they saw that the pellet was fluorescing green, it meant that the DNA has what had been inserted into the bacteria and not the protein. And so this conclusively showed that the genetic information that sent our traits from one generation to the next was indeed DNA. So we know that DNA is made up of a monomer called a nucleotide. DNA has what's called a double helix structure. So you can see on the picture on the left, this is the double helix. It's got these kind of backbones on the outside that are made up of alternating sugars and phosphates. And the sugar is called deoxyribose. And then in the center of these two backbones, you can see the nucleic acids, the adenines, thymines, cytosine, and guanine. And so DNA contains only 4 nucleic acids. We have thymine and cytosine are only made up of a single ring called pyrimidines. And then adenine and guanine are made up of 2 rings, and we call these purines. We know that adenine always pairs with thymine, and that guanine always pairs with cytosine. And this is a rule that the DNA follows, which allows it to keep the same length or width between the two sides of the backbone. It also allows it to have a really good mechanism for copying DNA. There is another molecule called RNA that we use for temporary genetic codes in the body, and this is also a nucleic acid. So RNA and DNA are very similar, except for a few key differences, which we've covered before. DNA is made up of a double helix structure, and RNA is only single-stranded usually. DNA contains the base thymine, right? So you can see the thymine here. Whereas RNA contains this base uracil. If you look, thymine and uracil are very similar, but you're never going to find uracil in DNA. And then the sugars are different. The DNA contains the sugar deoxyribose, which you can see on the left, and then RNA contains the sugar ribose. So up until the 1950s or so, we knew that DNA was the genetic material thanks to Hershey and Chase, but we didn't really know the structure of DNA until Watson and Crick and Rosalind Franklin came along. Watson and Crick and Rosalind Franklin were not working together. They were actually rivals. And Rosalind Franklin was using a technique called X-ray crystallography. So here's Rosalind Franklin, and you can see her famous X-ray crystallography picture of the DNA. And so what this was is she took a crystal that she made out of DNA, and she would shoot X-ray radiation through it, and it would project an image, and this is the image she got. And they could use this technique to figure out that DNA was a double helix, which was really the one clue that Watson and Crick needed to figure out the structure. You can see Watson and Crick here on the right with one of their original models, where they had these nucleic acid bases on the outside, which was wrong. And once they saw Rosalind Franklin's image, they were able to figure out the structure. Rosalind Franklin didn't actually give them this image willingly. It was on her desk, and an associate of hers gave it to Watson and Crick. So this was not done with her consent. And so Watson and Crick were given the credit for the discovery of the structure of DNA, and they received a Nobel Prize for it. Unfortunately for Rosalind, by the time they received this Nobel Prize, she had actually died. In the 50s, we didn't know that X-ray radiation caused cancer, and so she died of cancer from overexposure to radiation. But you can see down here in the picture, we have a couple of different images of what the DNA double helix looks like. In the photo on the right, you can see a computer-generated model, and so this has got all of the molecules on here. And again, you can see this outside backbone of sugars and phosphates, and then in between these backbones are the A's and T's and G's and C's all bound together. One thing to note about DNA, see how the phosphate is on the top on this side? But it's not, it's on the bottom on this side. DNA runs in a structure called anti-parallel. So the left side here is running from blue to yellow, blue to yellow, blue to yellow, blue to yellow. And then to get the same pattern on the right, it actually runs like that from the top down. So they run in opposite directions. And so we call this anti-parallel to show that they are facing opposite directions.